Okay, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. So today we are going to learn, okay, this time we are going to learn about the control of microbial growth. Okay, in previous lecture, we have discussed about the microbial growth, what factors contribute the microbial growth, okay, what are the stages. So in today's lecture, okay, what we are going to learn is about how we want to oppose, how we want to counteract the microbial growth, as we know that most microorganisms, okay, that we know could affect, could give a bad health effect to our human body system. So it is very important to control of this microbial growth so that we can prevent the disease or any infection, okay, from the cause of microorganism. Okay, right. So early civilizations practiced salting, smoking, pickling, drying, and exposure of food and clothing to sunlight to control microbial growth. So masa awal-awal dulu, okay, they have found methods to control of this microbial growth, such as salting, meletak garam, smoking ataupun kita panggil salai, atau menggunakan asap, pickling, drying, and exposure of food and clothing to sunlight. Okay, yang mana if we realize that all of these methods actually could affect the factors contribute to the microbial growth. So bila kita dapat tackle these factors, kita dapat counteract or oppose these factors, kita boleh menghalang this from prevent this microbial growth. Use of spices in cooking was to mask taste of spoiled food. Some spices prevented spoilage. In mid-1800s, okay, Semmelweis and Lister have developed aseptic, aseptic maknanya septic tu infection, aseptic maknanya tidak ada infection, aseptic techniques to prevent contamination of surgical wounds. Before then, nosocomial infection caused death in 10% of surgeries, up to 25% when the delivering in hospital died due to infection. So these two we call as a scientist, okay, Ms. Philip Semmelweis and Joseph Lister, okay, both contribute to the development of technique to control of the microbial growth using the antiseptic technique. And Semmelweis implemented hand antisepsis, okay, which cause reduction of microorganism on hands yang masih lagi kita guna, gunakan antiseptik, basuh tangan, okay, hand sanitizer, development of hand sanitizer. So this person actually responsible for the development of this antiseptic technique or aseptic technique. And Joseph Lister implemented wound antisepsis, precursor of skin antisepsis. Right, so that's a little bit about the overview of the control of microbial growth. So we are going to learn some definition for some terms, okay, such as sterilization. Sterilization is a cleaning or removing all forms of microbial life, including endospores, in a material or an object. Heating is the most commonly used method of sterilization. We also got commercial sterilization, which is a heat treatment, okay, menggunakan haba that kills endospores of Clostridium botulinum, the causative agent of botulism in canned food. Yang mana selalunya dalam makanan yang mempunyai dalam tin akan terdedah untuk mendapat kita panggil botulism toksin yang dihasilkan oleh Clostridium botulinum ni. Okay, so it is very bad, boleh menyebabkan what we call boleh menyebabkan penyakit terhadap penyakit disebabkan infection di dalam canned food ini. Okay, so heating, kita tahu setiap bakteria ni dia mempunyai certain temperature for their growth. So when we heat lebih tinggi daripada dia punya suitable temperature, we can kill the bacteria. So, however, it does not kill and dispose of the thermophiles which are not pathogens and may grow at temperatures above 45 degree. Yang mana yang boleh tahan, or we call can resist very high temperature. So this kind of what we call kind of technique to control the microbial growth cannot do that lah, tak boleh nak matikan and dispose of thermophiles yang boleh resist temperature, okay, and even they may grow at temperatures above 45 degree, okay. So what is disinfection? So disinfection is reducing the number of pathogenic microorganisms to the point where they no longer cause diseases. Okay, usually involves the removal of vegetative or non-endospore forming pathogens. So kita nak mengurangkan pathogenic microorganism yang mana bila bilangan dia terkurang, dia tidak boleh menyebabkan infection ataupun penyakit kepada or host tu lah. Okay, so it may use physical or chemical methods. Dia boleh menggunakan sama ada physical ataupun kimia. So such as disinfectant applied to inanimate objects, antiseptic applied to living tissues. Ni mechanical removal of most microbes in a limited area, example alcohol swab on skin, okay, degerming. Sanitization, use of chemical agent on food handling equipment to meet public health standards and minimize chances of disease transmission. For example, hot soap and water. Okay, so ini yang kita amalkan untuk salah satu kita amalkan seperti menggunakan disinfectant, sanitization, yang to sanitize tangan, especially with current COVID-19 situation. Okay, so this is what types of control of microbial growth. Okay. So what is sepsis? Sepsis comes from Greek for decay or putrid, indicates bacterial contamination. Asepsis menunjukkan absent. Sepsis menunjukkan contamination, so absence of significant contamination. Aseptic techniques are used to prevent contamination of surgical instruments, medical personnel and the patient during surgery. Aseptic techniques are also used to prevent bacterial contamination in food industry. So basically, aseptic technique ni sangat berguna, especially dalam keadaan yang mana memang kita tak nak ada pendedahan terhadap bakteria. Sebagai contoh dalam surgical punya operation theatre, yang mana bila kalau ada bakteria yang boleh hinggap, dia mudah masuk ke dalam badan. Bila kita operate means that we open our body, so inside the body's organs, tissues which exposed to the contamination will cause septicemia, bacteremia, yang mana akhirnya boleh lead to death. Okay, so it's very important this aseptic technique to be applied, especially in a condition like which mentioned here in surgery and also in food industry, because in food industry we will eat this foods stored in the can, okay, and it can also enter our body and cause contamination, bacteremia, sepsis and so on. So it is very important to apply aseptic technique in our daily life activities. Okay, so what is bacteriostatic agent? Okay, an agent that inhibits the growth of bacteria but doesn't necessarily kill them. Okay, stasis means to stop or steady. So bacteriostatic means stop bacteria but not kill, doesn't necessarily kill them. Maknanya dia menghalang atau memberhentikan, membantutkan pertumbuhan, inhibits the growth of bacteria, but it doesn't necessarily kill them. Okay, germicide, an agent that kills certain microorganism. So dia bila kita bactericide, kills bacteria lah. Viricide, inactivate viruses. Fungicide, agent that kills fungi. Sporicide, an agent that kills bacteria and the spores of fungal spores. Okay. Alright, rate of microbial death. When bacterial populations are heated or treated, antimicrobial chemicals atau apabila sesuatu bakteria tu di-expose kepada suhu yang sangat panas ataupun being treated by antimicrobial chemical, they usually die at a constant rate. Sama juga dia boleh growth in a constant rate. Bila kita apply sesuatu type of antimicrobial growth, dia juga boleh turun ataupun die at a constant rate. Okay, so one log decrease here causes 90% of population kill. Okay, ini low population load, ini high population load. By time kita boleh nampak bila dia semakin kurang dalam keadaan constant. Bila kita constant, kita boleh guna log ni mathematical formulation lah. So rate of microbial, there's several factors influence the effectiveness of antimicrobial treatment. Sama juga factors yang influence the growth of microbial, okay, microorganisms. So ada juga factors yang influence the effectiveness of antimicrobial treatment, such as number one, number of microbes. The more microbes present, the more time it takes to eliminate population. Semakin banyak microbes tu, so it takes time lah untuk eliminate semua sekali. Type of microbes. Endospores are very difficult to destroy. Vegetative pathogens vary widely in a susceptibility to different methods of microbial control. Okay. Environmental influences. Presence of organic material, blood, feces, saliva tends to inhibit antimicrobials, fish, etc. So, keadaan sekeliling juga memainkan peranan. Alright. Time of exposure. Chemical antimicrobials and radiation treatment are more effective at longer times. In heat treatments, longer exposure compensates for lower temperature. Okay, secara generalnya apabila kita expose makin lama, so dia akan lebih efektif lah. Okay, right. So physical method of microbial control, kita ada heat. Okay, heat kill microorganism by denaturing their enzymes and other proteins. Heat resistant varies widely among microbes. So certain microorganism, okay, usually they will be denatured lah disebabkan heat tersebut. Heat tu akan menyebabkan enzim dan protein dia kita panggil denature ataupun musnah lah. Okay, so basically we've got thermal death point. Okay, lowest temperatures at which all of the microbes in a liquid suspension will be killed in 10 minutes. Okay, thermal death time, minimal length of time in which all bacteria will be killed at a given temperature. Decimal reduction time, time in minutes at which 90% of bacteria at a given temperature will be killed, used in canning industry. Okay, thermal death point ni maknanya dia punya point lah, tahap dia punya kita panggil threshold ataupun temperature yang mana, okay, microbes ini boleh dibunuh dalam masa 10 minit. Thermal death time ni berapa lama masa yang diperlukan untuk bakteria ni mati dalam given temperature, while decimal reduction time masa yang mana 90% of bacteria will be killed. Okay, so tadi kita ada heat, ini moist heat kill microorganisms by coagulating their proteins. In general, moist heat is much more effective than dry heat. Okay, then we've got boiling heat up to 100 degrees Celsius or more at the sea level kills vegetative forms of bacterial pathogens, almost all viruses and fungi and their spores within 10 minutes or less. Endospores and some viruses are not destroyed this quickly, however, brief boiling will kill most pathogens. So, we've got example here, such as in hepatitis virus, can survive up to 30 minutes of boiling, and endospores can survive up to 20 hours or more of boiling. So, kita nampak ni, ada certain microorganism dia boleh survive dalam suhu yang tinggi ni dalam masa yang lama, such as yang mana ada endospores dan juga ada some type of viruses lah. Okay, but usually almost all virus dia akan mati dalam masa 10 minit or less when we use type of boiling. Alright. Okay, moist heat. Kita balik semula kepada moist heat. So what is moist heat? Reliable sterilization with moist heat requires temperatures above that of boiling water. Okay, kita ada contoh yang sangat common, which is what we call as the autoclave. So autoclave is a chamber which is filled with hot steam under pressure. Preferred method of sterilization unless material is damaged by heat, moisture or high pressure. So usually digunakan untuk one method of sterilization, okay, yang mana dia menggunakan proses hot steam under pressure. So temperature of steam reaches 1 to 1 degree of Celsius at twice atmospheric pressure. Most effective when organism contact steam directly or are contained in a small volume of liquid. All organism and endospores are killed within 15 minutes sahaja. Okay, berbanding dengan boiling tadi, alright, require more time to reach the center of solid or large volumes of liquid. Okay, so this is a picture of autoclave and closed chamber with high temperature and pressure, yang mana konsep autoclave ini adalah menggunakan heat, okay, dalam satu tekanan pressure yang tertentu, and it is different from the dry heat. It uses method of moist heat. Okay, alright. So another physical method, okay, yang kita nak belajar adalah pasteurization. It is developed by Louis Pasteur to prevent the spoilage of beverages. Is to reduce microbes responsible for spoilage of beer, milk, wine, and juices, etc. So kita ada classic method of pasteurization, yang mana contohnya susu, okay, will be exposed to 65 degree for 30 minutes, okay, and then high temperature short time pasteurization used today, yang mana dilakukan pada hari ini, will be exposed to 72 degree for 15 seconds, okay, and ultra high temperature pasteurization. Milk is treated at 1 degree for 3 seconds only and then cooled very quickly in a vacuum chamber. So pasteurization ni sangat-sangat kita selalu dengar susu pasteurized, yang mana baru kita tahu yang pasteurization adalah salah satu method untuk menghalang microorganism tu berada dalam minuman atau minuman susu tersebut lah. So the advantage of this method, pasteurization, milk can be stored at room temperature for several months. Okay, dia boleh kita panggil proses dia pun lebih cepat, and it can be retained in a longer time. Alright, and then we've got dry heat. Okay, dry heat ni kills by oxidation effects. Bagi contoh, kita ada direct flaming used to sterilize inoculating loops and needles. Heat metal until it has a red glow. Okay, maknanya kita direct bakar benda tu. Incineration, effective way to sterilize disposable items, paper, cups, dressing, and biological waste. And we've got also hot air sterilization, place objects in an oven. So basically, this kind of a method of microbial control require 2 hours at 170 degree for sterilization. Dry heat transfer heat less effectively to a cool body than moist heat. Okay, so basically kita ada dry heat, kita ada moist heat, what kind of method yang digunakan, guna application ataupun konsep yang sama. And then we've got filtration, removal of microbes by passage of a liquid or gas through a screen-like material with small pores. Used to sterilize heat-sensitive materials like vaccines, enzymes, antibiotics, and some culture media. So we've got two here, high efficiency particulate air filters HEPA, used in operating rooms and burn units to remove bacteria from air. HEPA ni selalunya kalau kita perasan dalam pembersihan iklan-iklan pembersihan udara, selalu ada yang mention this kind of terms lah, high efficiency particulate air filters. And then adik-adik kita ada membrane filters, a uniform pore size used in industry and research, ada different sizes, ada 0.22 and 0.45 micrometer pores. Used to filter most bacteria don't retain spirochetes, mycoplasma, and viruses. 0.01 micrometer pores retain all viruses and some large proteins. So there are different sizes with retain different type of microorganism based on their size lah. And then we've got low temperature. Low temperature maknanya dalam keadaan sejuk lah, ataupun beku. Effect depends on microbe and treatment applied. Kita ada refrigeration, yang mana selalu kita guna simpan makanan di dalam peti ais, temperature from 0 to 7 degrees Celsius. Bacteriostatic effect, they stop or inhibit the growth of the bacteria. reduces metabolic rate of the most microbes, so they cannot reproduce or produce toxins. Alright, and then we've got freezing, temperature below 7 degrees. We got two types of freezing, flash freezing and slow freezing. Flash freezing means that kita freeze dalam satu jangka masa yang cepat, but it does not kill most microbes. Okay, flash freezing is the process whereby objects are frozen in just a few hours. This is a keyword, few hours, by subjecting them to the cryogenic temperatures or through direct contact with liquid nitrogen at negative 196 degrees Celsius. It is commonly used in the food industry. Okay, flash freezing ni, nama pun flash means that the freezing operated in just a few hours. Okay, and then we've got slow freezing, more harmful because the ice crystals disturb cell structure. Over a third of vegetative bacteria may survive 1 year. Okay, in the previous freezing kind of freezing or type of freezing, in a flash freezing, it involves few hours, but yang ni boleh survive 1 year. Most parasites kill by a few days of freezing. Okay. And then we've got radiation. Okay, radiation ni kita ada beberapa banyak type. First sekali, ionizing radiation. Okay, such as gamma waves, X-rays, electron beams, or higher energy rays have short wavelength, less than 1 nanometer. Okay, and dislodge electrons from atoms and form ions, cause mutation in DNA and produce peroxides, use to sterilize pharmaceuticals and disposable medical supplies. Food industry is interested in using ionizing radiation. However, it got disadvantages such as penetrate human tissue, may cause genetic mutation in humans. Because yang kita tahu dia gunakan radiation lah, ionizing radiation. So there are side effects that need to be explained to the human kalau dia nak menerima radiation tersebut. Okay, alright. So basically kita ada banyak type of radiation. Ada gamma rays, X-rays, UV, okay. And then we've got visible light yang kita nampak hari-hari ni. And then we've got infrared, microwaves and radio waves. So basically these are kind of what we call radiation, and it depends on the dia punya wavelength dia lah. Okay, alright. Okay, ultraviolet light, which is a non-ionizing radiation, wavelength is longer than 1 nanometer, damages DNA by producing tiny diamonds which cause mutation. Used to disinfect operating rooms, nurseries, and cafeteria. Disadvantages: it damages skin, eyes, doesn't penetrate paper, glass, and clothes. Okay, ini guna cahaya UV lah. Alright. Okay. Alright, so now we go to the chemical methods of microbial control. So before this, we have learned about the physical and previous slides. Okay, so currently we go to the chemical methods lah. Okay, chemical kita ada types of disinfectant such as phenols and phenolics. Alright, so phenols and phenolics ni, phenol ni adalah carbonic acid, was first used by listed as a disinfectant. Rarely used today because it is skin irritant and it has strong odor. Used in some throat sprays and lozenges, acts as a local anesthetic. And then we got phenolics. Are chemical derivatives of phenol. Daripada phenol, kita boleh create these phenolics. Okay, so it is effective against gram-positive staphylococci and streptococci used in nurseries. Excessive use in infants may cause neurological damage. Okay, selalunya kita panggil bahan-bahan untuk buang ni lah, terdiri daripada phenolic atau phenol acid. So it destroy plasma membranes and denature proteins. Okay, so advantages: stable, persist for long times after applied and remain active in the presence of organic compound. So basically ini first chemical method yang digunakan lah untuk microbial control. Okay. And then we've got halogens. Halogens is effective alone on in compounds. Halogen yang pertama ada iodine. Iodine ni yang kalau kita luka ke, dia ada letak warna coklat. Dia punya cecair liquid tu. Okay, to enhance the recovery of the bleeding tu lah. So second method halogens such as A, iodine, Tincture of iodine alcohol solution was one of the first antiseptics used. Combines with amino acid tyrosine proteins and denatured proteins. Stain skins and clothes somewhat irritating. Okay, and iodophos compound with iodine that are slow releasing take several minutes to act as a skin antiseptic in surgery. Not effective against bacteria and the spores. Example is such as betadine and isodine. Dan ni kita selalu gunalah, especially bila kita nak buat surgery, prevention, selalu tempat yang kita nak potong tu, alright, dia akan letak dengan this iodine. And then we got chlorine. Okay, when mixed in water forms hypochlorous acid. Chlorine yang banyak kita dengar untuk membersihkan air kan. Alright, so bila dia campur dengan water, we call it as a hypochlorous acid. Used to disinfect drinking water, pools and sewage. Chlorine is easily inactivated by organic materials. Okay, sodium hypochlorite is active ingredient of bleach. Okay, yang mana untuk membunuh bakteria lah. Okay, so it is type of chlorine. So chloramines consists of chlorine and ammonia, less effective as germicides. Alcohol, okay, so alcohol biasa kita dengar penggunaan alcohol to kill bacteria, fungi but not endospores or naked viruses. Acts by denaturing proteins and disrupting cell membranes. Seperti mana kita dah belajar dalam prokaryotes, dia mempunyai proteins yang membentuk cell membrane ataupun dia punya structure tersebut. So alkohol ni it acts denature, maknanya dia pecahkan balik protein tu and also disrupt cell membrane. So this kind of organism tak boleh nak survive lah, right? Alcohol evaporate leaving no residue. Used to mechanically wipe microbes of skin before injections or blood drawing. Selalunya sebelum kita inject untuk keluarkan darah atau untuk ambil darah, selalunya kita akan swab dengan alcohol swab yang kita panggil itulah. So it is kind of alcohol. So not good for open wounds because cause protein to coagulate. For example, ethanol, drinking alcohol, optimal concentration is 70%, and isopropanol, rubbing alcohol better disinfectant than ethanol, also cheaper and less volatile, tidak mudah meruap. Okay. And then we got heavy metals as well, can be used as a chemical methods of microbial control, such include copper, selenium, mercury, silver and zinc. Oligodynamic action, very tiny amounts are effective. And kita ada like a silver, 1% silver nitrate used to protect infants against gonorrhea eye infections until recently. Ada mercury, organic milk compounds like methylate and mercurochrome are used to disinfect skin wounds, and copper. Copper sulfate is used to kill alga in pools and fish tanks. So these are examples of the heavy metal used to control the organism. And then we got selenium as well, because fungi and their spores used for fungal infection, also used in drop shampoos. And zinc chloride is used in mouthwashes. So basically kita dapat tengoklah banyak metal yang boleh digunakan untuk prevent this microbial control. Okay, and then we've got quaternary ammonium compounds, or we call it as a quartz. Okay, widely used surface active antigens, cationic or positively charged detergents, effective against gram-positive bacteria, less effective against gram-negative bacteria, also destroy fungi, amoeba and airborne viruses. Zafferan capable also found in lead sprays bottles. Pseudomonas strains that are resistant and can grow in presence of course are a big concern in hospital. Kadang-kadang sesetengah bakteria ni ataupun strain dia dah boleh resistant dekat certain chemical matter ni. So bahayanya resistant ni, maknanya tahap dia punya microorganism tu, dia punya defense, tahap defense dia tu tinggi, and it's very difficult to kill the bacteria, okay, using the current chemical methods yang kita ada. Okay, so penggunaan tu pun perlu betul. Okay, dan sometimes these prokaryotes they mutate and can also cause resistance, yang mana dia dah lali dengan bahan-bahan kimia tersebut. So advantages of quartz: strong antimicrobial action, colorless, odorless, tasteless, stable and non-toxic. However, the disadvantages are it forms foam, organic matter interface with effectiveness, neutralized by soaps and anionic. detergents. So yang seterusnya, next we've got aldehydes, include some of the most effective antimicrobials, inactivate proteins by forming covalent cross-link with several function groups. Kalau kita perasan, mostly chemical ni dia boleh bunuh dengan mengganggu sistem protein yang dia ada. Okay. Alright. So aldehydes ni, contoh ada formaldehyde gas, excellent disinfectant, commonly used as a formalin, 37% aqueous solution. Formalin was used extensively to preserve biological specimens and inactivate viruses and bacteria in vaccines. Irritates mucous membrane, strong odor, also used in mortuaries for embalming. Formaldehyde ni selalunya atau digunakan formalin selalu kita guna dalam pengawetan mayat ke kalau untuk student-student medik untuk nak belajar anatomi daripada mayat tu kita panggil di approach ataupun dimasuki atau invade by the microorganism. So formaldehyde gas ni salah satu yang selalu digunakan lah as kita panggil pengawetan mayat. Okay. And then we've got glutaraldehyde, less irritating and more effective than formaldehyde, one of the few chemical disinfectant that is sterilizing agent. 2% solution of glutaraldehyde oxidates is bactericidal, tuberculocidal, dibunuh tuberculosis juga, and virucidal in 10 minutes. Spirocidal in 3 to 10 hours, commonly used to disinfect hospital instruments, also used in mortuaries for embalming. And then we've got gases sterilizers, okay, chemicals that sterilize in chambers similar to an autoclave, denature protein by replacing functional groups with alkyl groups. Sebagai contoh kita ada ethylene oxide, okay, kill all microbes and endospores but require exposure of 4 to 18 hours. Okay, toxic and explosives in pure form, highly penetrating. Most hospital has ethylene oxide chambers to sterilize mattresses and large equipment. Alright, and then we've got peroxygens or oxidizing agents. Okay, peroxides yang boleh menyebabkan oxidization. Okay, oxidize cellular components of treated microbes, disrupt membranes and protein. Kita ada ozone, used along with chlorine to disinfect water, help neutralize unpleasant taste and odors, more effective killing agent than chlorine but less stable and more expensive. Highly reactive form of oxygen, and by exposing oxygen to electricity or UV light. Oxygen ni seperti yang mana kita tahu selalunya sangat berguna, very beneficial to our body, but when it is oxidized, it can be a radical, kita panggil radical substance yang boleh menyebabkan benda tu jadi berbahaya kepada badan kita pula. Okay, so ozone ni dia adalah highly reactive form of oxygen. Sebagai ROS reactive oxygen species yang mana dia boleh menyebabkan kemusnahan sel. Okay, so ini yang digunakan oleh ozone. And then we've got hydrogen peroxide, used as an antiseptic, not good for open wounds because quickly broken down by catalysts present in human cells, effective in disinfection of inanimate objects, poricidal at high temperatures, used by food industry and to disinfect contact lenses. Kita ada benzoyl peroxide yang kita gunakan untuk buang jerawat. Okay, and then we got peracetic acid, one of the most effective liquid sporicides available. Sterilants kills bacteria and fungi in less than 5 minutes, and the spores and viruses within 30 minutes. Is widely in disinfection of food and medical instruments because it does not leave toxic residue. Alright, okay, so ini adalah kita boleh katakan in summary type of antimicrobial growth yang mana dia punya percentage of bacteria on how it could affect the percentage of the bacteria surviving and how long it takes to decrease this bacteria surviving percentage lah. Yang mana soap and water, it takes longer time. And then we've got aqueous zephyrin, mixture of 50% ethanol and acetone, tinctures of zephyrin, 70% ethanol and 30% of water, and 1% iodine is 70% ethanol. Okay, tincture of iodine adalah yang paling cepat, yang mana hanya memerlukan 90 seconds to increase the bacteria of surviving significantly. Alright. Okay, I think that is uh, the final slides, the final slide for this lecture. So hopefully all of you can understand this topic. This is quite direct. Okay, no need to there are things or terms that need to be memorized, kena benda-benda kena hafal. Tapi saya rasa tak mudah untuk tak mudah pula, tak adalah sukar untuk difahami. It's very easy to understand this topic. Okay, so just have to remember some to memorize to some words, terms, like types of antimicrobial growth. Okay, so I think that's all. Thank you very much for listening this lecture. Okay, thank you.